Welcome everyone to this presentation on immune cellular therapy of cancer with emphasis on new strategies in the cellular therapy of solid tumors. I'm Samuel Bernal, and I would like to acknowledge my colleagues at Globe Tech Science Foundation in the Philippines, University of California in Los Angeles, Cedars-Sinai Medical Center in Beverly Hills, California, and the Institute of Personalized Molecular Medicine at the Medical City in the Philippines. The topics we are going to cover today include the types of immune cells for therapy of cancer, constructs of cellular therapies with targeted receptors used in the treatment of cancer, strategies to optimize immune cell therapies for cancer, both in vitro and in vivo, the experience of immune cell therapies in the Philippines, future directions in cellular therapy of cancer, and the summary. The types of immune cells for therapy of cancer include dendritic cells, cytokine-induced killer cells, receptor CIK and chimeric antigen receptor T cells, natural killer cell-based therapies, and gamma delta T cells. I'm going to discuss each one of them in just a brief outline and present some applications. Dendritic cell therapy is vital for immunosurveillance, and dendritic cell allows the recognition of the immune system of specific antigens in malignant cells. They are stimulated ex vivo and loaded with tumor-specific antigens that are dependent on the major histocompatibility complex, with the intent of activating specific T cells to selectively eliminate those antigen-bearing cancer cells. There are limitations in terms of the expression of inhibitory signals of the tumor, and the MHC may not be expressed in the tumor or would be downregulated, and the high tumor load induces a tumor-induced immune suppression, including those mediated by T regulatory cells and myeloid-derived suppressor cells. To emphasize the role of dendritic cells, even in the use of immune checkpoint inhibitors, recognizing that, of course, the effects of immune checkpoint inhibitors would depend upon their interaction with immune cells, and dendritic cells play a critical role. Even in the use of immune checkpoint inhibitors such as the anti-PD-L1 or anti-PD-1, the anti-tumor responses require interleukin-12 producing dendritic cells for their action. The PD-L1 PD-1 blockade stimulates the CD8 T cells to produce interferon gamma, which then activates dendritic cell production of interleukin-12. The NF-κB pathway enhances dendritic cell interleukin-12 production, and combining then the checkpoint inhibitors with dendritic cells could enhance checkpoint immunotherapy. The combination strategies to maximize the therapeutic effectiveness of dendritic cells include ways to exploit the intrinsic capacity of dendritic cells to stimulate anti-tumor immune effector cells, such as tumor antigen-specific cytotoxic T lymphocytes and natural killer cells. So, therapeutic interventions to enhance the strength of immune effector response include reversal of the tumor-associated immunosuppression, to reduce tumor burden and increase the immune susceptibility of tumor cells, and the use of combinations such as dendritic cells with cytotoxic T cells, cytokine-induced killer cells, and natural killer cells. Clinical responses to dendritic cell and dendritic cell combinations have been shown in melanoma, with a modest objective response when dendritic cells are used alone, with similar efficacy to standard treatments such as dacarbazine or ipilimumab. For prostate cancer, dendritic cell therapy has been approved by the US FDA based upon improvement of overall survival in the experimental group superior to what was observed in the control group. In patients with malignant glioma, dendritic cells have also shown an objective response of about 15%. In advanced renal cancer shows an objective response of about 11%. And in hepatocellular carcinoma, the combination of dendritic cells and cytokine-induced killer cells with TACE have shown improvements in half-year, 1 year, and 2-year median overall survival and progression-free survival in hepatocellular cancer patients compared to TACE alone. Now, cytokine-induced killer cells have also been used, which are non-MHC restricted T cells, taking advantage of the body's natural ability to eliminate tumor cells. They are a heterogeneous group that contain a high percentage of cells with a mixed T cell and natural killer cell phenotype. So the majority of CIK cells express T cell receptors which have a high proliferation rate, high cytolytic activity, and non-MHC restricted killing of tumor cells through activation of the natural killer group 2 member D or NKG2D cell surface receptor. And clinical trials have demonstrated the feasibility and efficacy of CIK immunotherapy even in advanced stage cancer patients or those that have not responded to first-line treatment. The generation of cytokine-induced killer cells go through leukapheresis, culture, stimulation with anti-CD3 in interleukin-2, and then expansion in culture, and eventually infused to the patient. And the in vivo activity, like natural killer cells, also use the NKG2D receptors that interact with their ligands, and cytotoxic activity is triggered by the lysis of tumor cells to the release of perforin and granzyme granules, and exert also anti-tumor activity by the ADCC mechanism. Clinical responses to dendritic cell and dendritic cell cytokine-induced killer combinations have been described in tumors, including those that have already failed prior therapy or compared to other standard therapies, as was observed in hepatocellular carcinoma patients, also observed in those patients with different types of advanced tumors, and the autologous cytokine-induced killer cell immunotherapy have shown longer progression-free survival compared to the control group. The third type that we are going to discuss are those that are receptor-engineered immune cells, including CAR-T and other CAR immune cells that function by redirecting the immune cells to a predefined tumor-specific target. And the antigen-binding moieties could include the SCFV domain derived from antibodies, the human FAB fragment, and ligands that engage their particular cognate receptor. The fourth type are natural killer cells, which are innate lymphoid cells that can induce the death of allogeneic and autologous cells undergoing malignant transformation. They express an array of activating receptors such as NKG2A and NKG2D on the cell surface. In patients with advanced renal cancer, some trials have already shown efficacy even with allogeneic natural killer cells, as well as the CAR-engineered NK cells studied in B-cell malignancies, breast and ovarian cancer, colon cancer, prostate cancer, and sarcoma. One advantage of natural killer cells compared to total T cells, including those used in CAR-NK constructs, are that mature NK cells are short-lived, and are expected to disappear rapidly after mediating their anti-cancer efforts. And this is the advantage of less toxicity from prolonged cytokine release syndrome and toxicity against normal cells compared to total T cells. The fifth type, which we are just briefly going to cover, are the gamma delta T cells. We have a T cell receptor composed of one gamma chain and one delta chain, even though they share many qualities with their more common alpha beta chain counterparts, with cytotoxic effector functions, pro-inflammatory cytokine production, but the major difference is that they are independent of the major histocompatibility complex molecules. They do not seem to require antigen processing in order to present the peptide episodes, but some recognize the MHC class 1b molecules, and they are believed to have a prominent role, particularly in the recognition of lipid antigens. The mechanisms by which the gamma delta T cells kill cancer cells are similar to cytotoxic T cells, and also involve the release of perforin and granzyme B, as well as the tumor necrosis factor related apoptosis inducing ligand and fast ligand inducing apoptosis in the target cancer cells. The second topic are the constructs of cellular therapies with targeted receptors, including the generation of CAR and armored T cells. There are several generations of these CAR constructs, and the first generation include activating receptors, with the second generation combining both activating receptors and co-stimulatory signals such as CD28. The third generation CARs combined two co-stimulatory and activating signals such as 4-1BB, and the fourth generation CAR T cells, which is the type that we are using in our own laboratory, sometimes called TRuC cells, can secrete cytokines upon the activation of the CARs. They recognize tumor cells through their tumor-associated antigen in a non-MHC restrictive manner, then secrete cytokines that kill tumor cells and induce them to attack other tumor cells. Briefly covering those CAR T cells approved for hematological cancers, including Yescarta for the treatment of large B-cell lymphoma, the Trisatgin Lucil, FDA-approved CAR T-cell therapy for diffuse B-cell lymphoma, and the Brexucabtagin CAR T construct used for relapse or refractory mantle cell lymphoma. And all of these have shown very good results. The US FDA has approved this treatment for patients who have not responded to standard therapy or who have relapsed following 2 or more lines of systemic therapy in diffuse large B-cell lymphoma, mediastinal B-cell lymphoma, high-grade B-cell lymphoma, and DLBCL type of lymphomas. The other CAR T constructs, including Tisagenlecleucel, which is approved for refractory diffuse large B-cell lymphoma, and Brexucabtagin, which was the first gene therapy to be approved for the treatment of relapsed or refractory mantle cell lymphoma. All of these CAR constructs in hematological cancers have shown very dramatic responses, with 90% complete remission in patients with refractory or relapsed ALL, 80% overall clinical response in patients with advanced multiple myeloma, about 57% response in patients with relapsed or refractory CLL, and 53% overall response in patients with advanced lymphoma. So even though the responses for CAR T in hematological cancers is clear and very dramatic, the focus of this presentation now is the more challenging target, which is solid tumors. Some of the strategies to optimize immune cell therapies in vitro and in vivo. This presentation is based upon a paper that we had published on CAR-T therapy for solid tumors, including the development of new strategies. And particularly the major challenge in solid tumors, the generation of novel CAR constructs for the different solid tumor types, representing some of the new technologies that we developed in our own lab to enhance efficacy while decreasing the toxicity of CAR-T therapies in solid tumors. The challenges include the identification of optimal tumor antigens, trafficking of the adoptive transferred cells to tumor and metastatic sites, and very important in solid tumors is the neutralization of the immunosuppressive tumor microenvironment. Some of the technologies to optimize efficacy of cell therapies in solid tumors include the characterization of the immune cells by mass cytometry and single cell RNA sequencing, targeted augmentation or inhibition of cell receptors to make the cellular immunotherapy more effective, and knockdown of inhibitory receptors using CRISPR to augment their anti-tumor function. Some of the strategies for improvement of CAR-T are directed to the microenvironment in the tumors that distinguishes them from many of the hematological cancers. And this is just an overall graphic of the numerous factors that have to be taken into account, which is why the immune cell therapy for solid tumors presents a major challenge. But there are strategies that have been developed, and many of these strategies have already shown good efficacy in solid tumors. Some of these strategies involve the CAR-T cell trafficking, homing and extravasation of the CAR-T cells to the tumor site, infiltration into the tumor microenvironment, producing and potentiating the tumor targeting by fine-tuning the scFv affinity, neutralizing CAR-T toxicity, targeting the heterogeneous population of cancer cells, evading the complexity of genomic instability, bypassing immune checkpoint inhibition, circumventing downregulation of antigen targets, and using the molecular profiling of tumors to identify known and neoantigens. So this is a graphic of the differences between the first generation, second generation, third generation, and the fourth generation, which is the strategy that we use, that includes the interactions between the CAR constructs with dendritic cells, macrophages, and other immune cells, including natural killer and gamma delta T cells. So these fourth generation CAR T cells we are able to combine with the effector cells, including dendritic cells and the effector T cells, as well as NK cells and gamma delta T cells. The CAR T preparation goes through leukapheresis with production of the vector, transduction, expansion, and culture, and finally dosages for administration into patients, carefully monitoring their toxicity. Now, the advantages of CAR T cells in immunotherapy of tumors is that CAR T cells have the ability to recognize antigens regardless of the human leukocyte antigen background, whereas natural T cell receptors have to be expressed on the T cell surface that need to be matched with the patient's haplotype. And HLA expression may be downregulated in some tumor cells, but CAR T cells are still able to eradicate these escaping tumor cells, and CAR T cells have a wider range of potential targets because they not only include proteins but also carbohydrates, glycoproteins, and glycolipids that could be major tumor targets. So CAR compared to T cell receptor mechanisms are HLA independent. They have higher bonding affinity. There is a direct correlation between proliferation and treatment response, and but in common with the T cell receptor are also engineered for introduction of recognition of tumor-associated antigens. The cloning and construction of the CAR plasmid vector is quite complex when targeted against solid tumors, particularly with the identification of a heterogeneous group of subclones of the solid tumor that may require different types of target antigens, the plasmid propagation, isolation, electroporation, and mechanisms of monitoring cell viability, the CAR expression and amplification, and confirmation of copy number levels. These are some of the constructs that we have used to be able to develop anti-WT1 constructs of CAR T cells that uses a vector to be able to express the CAR receptor without the use of replicating viruses. DNA sequencing is a critical part of the confirmation of these different constructs, which we have done with different constructs, WT1 directed and CD19 CAR constructs developed in our laboratory. WT1 CAR constructs are especially interesting to us because they are expressed in many malignancies, including various hematopoietic cancers, but solid tumors also express or overexpress WT1, including lung cancer, melanoma, mesothelioma, and many other solid tumors. Now, in the case of mesothelioma, a CAR T derived targeted against mesothelin was found at Memorial Sloan Kettering to have had a very strong overall response in when administered into the pleural cavity, and these CAR T cells persisted in the pleural fluid for several weeks. But this treatment was combined with an immune checkpoint inhibitor, in this case pembrolizumab. So the projects in our laboratory, as I mentioned, include WT1 construct, anti-mesothelin, anti-CD19, CD30, CD20, combination of bispecific WT1 and CD19, particularly directed to malignancies that downregulate CD19, anti-CD25, anti-PSMA for prostate cancer, and anti-EGFR. To confirm the activity, or to be able to fine-tune the interaction of these CAR constructs with the target tumor cells, we developed an in vitro assay which uses a cellular retention to be able to assess and quantify the interaction of the CAR receptor engineered cells against the target tumor cells, in this case CD19 positive leukemic cells, but they could be applied to solid tumor cell lines in vitro as well. So the cellular retention model is described here, which uses the CAR construct with the receptors in the target cells, and followed by washing and quantitation of the bound CAR engineered cells to the target cells immobilized on a matrix. And this allows us to be able to show that the level of the amplification of the CAR receptor is also related to the interaction of the CAR constructs to the target cells, and the profile of the expression of the construct in the immune cells are also quantitated and show a correlation with their affinity and their level of interaction with the target cells. This can also be confirmed by microscopy to be able to see the specificity of the interaction of the CAR immune cell construct with the specific antigens expressed on the target tumor cells. And this describes all the controls to be able to identify specificity and effectiveness of the binding of the CAR constructs to the target cells. The experience of immune cell treatments for cancer in the Philippines, particularly focus in the programs we have at Medical City, as well as Lung Center of the Philippines, include a large variety of solid tumors, particularly lung, breast, ovary, and also head and neck and prostate, but also a variety of hematological cancers, including lymphoma, ALL, AML, CLL, Hodgkin's lymphoma, and multiple myeloma. In the use of dendritic cells, we published many years ago with our colleagues, including particularly Dr. Ona, who was the former Secretary of Health, and Dr. Gloria Crista Luna, who is an oncologist and a sponsor of this conference, identifying the anti-cancer immune reactivity and the long-term survival after treatment of metastatic ovarian cancer with dendritic cells. And this happens to be a 35-year-old doctor who initially presented with unknown primary. Later on, we determined, based upon markers, to be of papillary type, or with ovarian features, showing metastasis of the tumor in the lungs, in the liver, and primary tumor and extension into the pelvic organs, which increased after this patient failed multiple courses of chemotherapy. She was then treated with dendritic cells targeted to specific antigens of her own tumor, and over a period of 2 years there was complete resolution of her lung metastasis, as well as complete resolution of her liver metastasis and the disappearance of the tumors in the pelvic area over a period of 2 years. This was already in 2006, and up till 2020, at the current time, she is still in complete remission. So this is a patient who had already failed multiple courses of chemotherapy and unable to tolerate more chemotherapy because of severe neuropathy and thrombocytopenia, and even with the use of dendritic cells alone, she was able to achieve a complete remission. But the construct of the dendritic cells and the engineering of the dendritic cells to recognize the specific antigens of her own tumor, personalized immune cell therapy targeting was critical for the success of this treatment. I would just like to have a brief overview of what we feel are the future directions of cellular therapy, including the combinations of immune cell therapy with immune checkpoint inhibitors, combinations with interacting immune cells, aside from total T cells, would be natural killer cells, which also play a pivotal role, and also cytokine-induced killer cells and gamma delta T cells. There are continuing efforts to enhance the efficacy of CAR constructs specific for solid tumors, but because of the complexity, there is immune cell targeting of the cancer cells from our understanding of the complexity of the tumor microenvironment, particularly in solid tumors, and understanding the interaction between cancer cells and immune cells that we were able to model in vitro, and making extrapolations of their activities in vivo, and to strategies to target multiple mechanisms by which cancer cells can suppress the anti-tumor function of innate and adaptive immune cells. I wish to emphasize that what we are using now for patients is really beyond CAR-T, because we do not use total T cells unlike the traditional CAR-T. so that the more advanced programs allow us to be able to enhance anti-tumor activity while decreasing the risk of toxicity. So we do not use viral vectors that are replicating because of the concern of genotoxicity as a result of the integration into unintended locations in the genome and replication of these viral vectors, particularly in using total T cells that may persist in the body. So instead of the replicating viral vectors, we use non-viral vectors, but still are able to achieve high expression of the receptors in the surfaces of the immune cells. So another distinguishing point is that the adaptive immune cell transfer program that we create is personalized for each patient based upon the molecular characteristics of that patient's tumor. So there is no one size fits all, and these are more characterized as perhaps akin to stem cell transplants rather than the analogy of mass-produced drugs. We are giving more emphasis on natural killer cells as part of the innate immune system, which have roles also both in the innate and adaptive responses with unique lymphocyte subsets, and cell types can respond to the presence of tumor cells and participate in immune surveillance. Whereas the majority of the traditional CAR-T have focused on the total expression of T cells, the functional heterogeneity limits their therapeutic potential and is associated with toxicity. And we use more and more the receptor-modified NK cells to provide a method to direct these cells more specifically to the target cancer cells with less risk of adverse effects. So the ex vivo stimulation and expansion of these lymphocytes followed by reinfusion follows similar to what is used in CAR constructs, but the use of non-viral vectors as well as the use of other cell types other than total T cells is a major advantage, where we have observed not only greater efficacy and binding to specific receptors in the tumor cells, but also less common or less severe cytokine release syndromes were observed. NK cells are particularly interesting because they have spontaneous and innate cytotoxic activity and can trigger target cell death independent of the tumor antigen. And NK cells would still be effective against tumor cells even with antigen downregulation. And ex vivo expansion of this human NK cells to produce interferon gamma, interleukin 3, and GM-CSF that differ from the pro-inflammatory cytokines that may be produced simultaneously by T cells and associated with toxicity associated with cytokine release syndrome, which may explain why NK cells targeted against specific tumor antigens may be less toxic than total T cells. And instead of total T cells, which could persist and may be associated with a higher toxicity, particularly targeting normal cells and associated with a higher cytokine release syndrome, we use cytokine-induced killer cells, NK cells, and gamma T cells to induce greater but more specific anti-tumor action. The unique ability of natural killer cells make them ideal candidates for use in solid tumors with the use of cytokine and cytokine analogs, blocking the killer cell immunoglobulin-like receptor or KIR interactions with anti-KIR monoclonal antibodies, preventing CD16 receptor shedding. They use immunocytokines and immune checkpoint inhibitors that can enhance NK cell-mediated cytotoxicity, and methods to redirect the NK cells to recognize and target specific tumor-associated antigens. In summary, immune cell therapies have revolutionized the treatments of cancer, starting with the clear and dramatic responses with hematological cancers, but now also extended to solid tumors. There is an often quoted dictum that immune cell therapies, including CAR-T, do not work in solid tumors. This is rapidly being challenged by development of new strategies of immune cell therapies directed to specific solid tumors, particularly when they are personalized and we are able to confirm their affinity, specificity, and specific targeting to specific antigens of a heterogeneous group of tumors. There are numerous factors that could affect response of solid tumors to immune cell therapies, which presents a major challenge, but the technology has advanced so that we are able to address many of these factors so that immune cell therapies are finding their place in the treatment of solid tumors, particularly dealing with heterogeneous expression of antigens in solid tumors and expression of these molecular markers, particularly with their biological behavior and their development of treatment resistance. No two patients react or respond to cancer treatments in the same way. So the one-size-fits-all approach, particularly directed to immune cell therapies in solid tumors, do not work. So the treatments have to be personalized. We have to take into account subpopulations of cells within the tumor with the properties of cancer stem cells. And the immune system is clearly critical in controlling cancer, and particularly by evading immune surveillance, and can actively suppress immune recognition. But preparative chemotherapy can also decrease T regulatory cells and allow better immune recognition of cancer cells in vivo. And the use of other immune cells such as dendritic cells, cytokine-induced killer cells, CAR T cells, and NK cells, as well as gamma delta cells that can be used to target specific antigenic profile of the tumor of each patient, can be engineered to be recognized with high affinity and confirmed in vitro as well as in vivo. Personalized combined molecular targeted therapies makes molecular profiling of the specific targets in each patient's tumor critical to analyze the evasions of the strategies used by the cancer cells to evade the immune surveillance, including the molecular mechanisms of drug resistance, and best used as an early modality to avoid this chemo or radiation burned out. Immune cell therapies are not really a treatment of last resort. And to balance safety, tolerability, and stem cell therapies, toxic effects can be modulated, as well as when we combine or we alternate the use of immune cell therapies with chemotherapy and radiation, we are actually observing the modulation of many of the toxic effects of chemotherapy or radiation, and overcoming drug resistance in the treatment of solid tumors by using mechanisms that are independent of the molecular markers that may be expressed by tumor cells, leading to resistance to specific drugs. But using immune cell therapies allows for an independent mechanism to be able to target even those drug resistant cells. And immune cell therapies can be used together with immune checkpoint inhibitors such as anti-PD-1 and anti-PD-L1 antibodies. There is a considerable amount of fundamental molecular biology to identify these molecular targets, neoantigens using genomics, proteomics, and metabolomics, and engineering of the human immune cells to be able to target the specific molecules of each patient's tumor, to monitor the specific immune recognition, to customize and adjust treatment strategies for each patient, and continuous interaction between scientists and doctors on one hand and the patient on the other throughout the treatment process. And a major block still in the widespread adoption of these therapies is, of course, the high cost of these treatments. And there are mechanisms that are being introduced to be able to help finance these new cellular treatments for greater access by patients. Thank you, and I hope great success for this conference as a whole.